myself Dr. Deepika Saini, Associate Professor at Lloyd Institute of Management and Technology. In the previous part of anticonvulsant drug, though we have covered a number of classes, and today we are going to discuss the remaining classes of anticonvulsant drugs. So, this is the drug classification. Just to just as a revision, we can say, like we have done the barbiturates, the phenobarbital and methabarbital, hydantoin, phenytoin, mephitoin and ethotoin, oxazolidine-diones, trimethadione was the drug that was the first introduced drug for the absence seizures, and paramethadione, succinimide, we did the synthesis of ethosuximide as well. Further, the urea and monoacyl ureas, we have studied about the structure activity relationship of urea and monoacyl ureas. Apart from that, we did the synthesis of carbamazepine. So, we are left with two different categories. One is benzodiazepine and the miscellaneous categories. So, let us begin with benzodiazepine. To begin with benzodiazepine, first we must know about what are benzodiazepine, as we have already studied it in the antipsychotic category. So, these are benzo means benzene, di means 2, aza means nitrogen, apine means 7-membered ring. So, we have some clues that it must contain a benzene ring, it must contain 2 nitrogen atom in 7-membered ring. So, we can say chemically, if we talk about as a structure, benzodiazepine is a fusion of benzene ring, as we have discussed, and a 7-membered diazepine ring. 7-membered diazepine rings means it contains 2 nitrogen atom. So, again we can clearly see this is the benzene ring, this is the ring A, and this ring B, that is diazepine ring, and in this we have 2 nitrogen atom. When we start numbering it, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. As you have as we have already studied about the numbering of this fused system, we know we always left this fusion phase, and at last we number it 10 and 11. At position 5, there is already a substitution, that is a benzene ring. So, what we did? We have substituted it with, we have shown the numbering with dash, 1 dash, 2 dash, 3 dash, 4 dash, 5 dash and 6 dash. As we have mentioned, we have substitution at this position R1, we can have substitution at this position R2 dash rather, and this R7 position. So, all these derivatives that are benzodiazepine, they are very well known for lowering the brain activity. They decrease the excitement, they decrease the neural transmission to produce any kind of activity, that is brain activity. So, thus they are used in several conditions such as anxiety, they are well known in case of anti-anxiety, insomnia. Even insomnia is a state when a person is not able to sleep, and so that condition is known as insomnia. And apart from that, seizures that we are talking about, that it is one of the best anticonvulsant drug. So, this is the example. Every benzodiazepine is not going to work against convulsions, though there are a selective compounds under the category of benzodiazepines that acts against convulsions or acts as anticonvulsant. So, we are discussing this 1, 2, 3, 4, 5, 6, 7, 8, 9. This is the numbering of this fused system, this benzodiazepine structure, and at position number 5 we have a substitution, we have a benzene ring. Again we are repeating it like 1 dash 2 dash 3 dash 4 dash 5 dash 6 dash, and this position is R2 dash, and this at this position number 7 we have R7 at position number 1 with the substitution of nitrogen. If we would say that we have R1. So when we talk about diazepam, so what happened here is we have substituted this position with a methyl ring, this with a chlorine, and there is no substitution at this benzene ring. So there are some structural examples of benzodiazepine, though all the structures, all the compounds under benzodiazepine category does not have the anticonvulsant potential, but some of them have been proven as better anticonvulsant compounds. So let's start with diazepam. So when we talk about the numbering, as we have already mentioned, this 1, 2, 3, 4, 5, 6, 7, 8, 9, and at position number 5 already there is a benzene ring, and this benzene ring is further when we talk about the numbering, that is 1 dash 2 dash 3 dash 4 dash 5 dash 6 dash, and the position this is R2 dash. We have substitution at R7, we have substitution at nitrogen number 1 as R1. So in case of diazepam, what we did, we have substituted this nitrogen with a methyl, and this R7 with the chlorine, and this R2 dash, that is this benzene ring that remain unsubstituted. In case of nitrazepam, we have not substituted nitrogen, we have not substituted the benzene ring. The only fused benzene that at position number R7 we have a nitro group. So this is nitrazepam. Then we have clonazepam also. In this case, all the things are almost similar to the nitrazepam. The only point is this R2 dash position, that is Cl in case of clonazepam. So when we talk about structure activity relationship of benzodiazepine, benzodiazepine has a very diverse structure when we talk about the structure activity relationship of benzodiazepine. So we have several targets in this case. The first target is nitrogen that can be substituted or not. Apart from that, we have a position number 5 which is being substituted with a phenyl ring. Further, this position at position number 7, that is being found with several substitutions, and then the position number 2 of the of a phenyl ring which itself act as a substitution for the position number 5. So we are going to study about these 4 main targets. So let's begin with the nitrogen one, the first one. So this N-methyl substitution that will increase the activity. So when we talk about this, let's see the if we talk about the diazepam, if we have not substituted this R1, this the substitution at position number 1 of nitrogen, if we have not substituted it with CH3, the potency of diazepam might be less. So we have found that the position when this substitution at nitrogen at the position number 1, nitrogen at position number 1 is being substituted, that has led to the more potent, active anticonvulsant agents. After that, we have this position number 5. A phenyl group at this position is essential for activity. So this phenyl, rather it is being substituted or unsubstituted, that is another part. But this position itself, the position number 5, must be substituted with a phenyl group. So, the two things that we have studied yet, the nitrogen that must be substituted to increase the activity, to enhance the activity. Though those unsubstituted nitrogen atoms, they also possess anticonvulsant activity, but it has been shown that the substituted one will possess much better activity as compared to the unsubstituted one. So, and at the position number 5, as we have seen, there is a already there is a substitution, that is a phenyl ring, that is must for the activity, that is required for the anticonvulsant potential. So, now when we talk about the substitution at this position R2 dash, so when we have substituted at ortho position with a halogen atom, that increases the activity. But when we have replaced this with any of the electron donating group, like suppose if we would say CH3, a methyl group, any alkyl group, that decreases the activity. So, we came to know about the fact that this R2 dash position, the second position of the substitution of phenyl substitute of phenyl ring as a substitution, that must be substituted with a halogen, and most preferably it has been seen the chlorine is the atom that is being used as the substitution at this position. When we talk about the position number 7, this R7, we have seen an electron withdrawing group at position number 7 that serves the purpose, that will increase the activity. But when we have seen, we have replaced this electron withdrawing group with an electron donating group that has decreased the activity, that lead to decreases the activity. So, it is very clearly very evident for this group, this benzodiazepine group, we have to make sure the presence of electron withdrawing group rather than electron donating group, both at position number R2 dash as well as at position number R7. So, when we talk about structure activity relationship of benzodiazepine, we have 4 different target. First one is the nitrogen at first position, then the second one is the phenyl, the position number 5 of diazepine ring, benzodiazepine ring. So, and the last one and the next one is the R2 dash position of phenyl ring, that is being substitution as a substitution at position number 5, and then R7 position of the fused benzene ring. So, when we talk about the nitrogen, the N-methylation, that definitely increase the activity. At position number 5, we have seen the phenyl substitution, it is essential for the activity. Apart from that, when we talk about the R2 dash position, we have seen the halogen incorporation has increased the activity, while the electron donating group like CH3 or any other alkyl group or electron donating group, and that has decreased the activity. When we talk about the R7, the scenario is almost similar. The electron withdrawing group that found to possess better activity, and while substitution with the electron donating group that has failed the substitution process, means that decreases the activity. There are some of the miscellaneous agents, the primidone, the valproic acid, the felbamate and the gabapentine. So, these are the important miscellaneous agents in which we can clearly see we have 2 ketonic group and the 2 NH group in the case of primidone. These are most essential part of this structure. And then we have valproic acid, we have a further we will look into the structure of valproic acid. We have felbamate, in which we can clearly see we have COONH2 group, we have CONH2 group there also, and in case of gabapentine we have COOH, that is carboxylic group is present in this drug. So, these are the example of miscellaneous agents: primidone, valproic acid, felbamate and gabapentine. These are the structural isomers of valproic acid. The valproic acid is also known as 2-propyl pentanoic acid. How? So, let us begin with it is a pentanoic acid, means it contains carboxylic group. So, we have numbered this carbon atom as 1, then the next one is 2, 3, 4 and 5. So, valproic acid is also at this 2 position we have CH2, CH2, CH3. So, it is a propyl group. So, at second position we have propyl group. So, it is known as 2-propyl pentanoic acid. It is known as valproic acid. When we have then we have another option that we can have this 1, 2 and at position number we have just divided these methyl group at 2 at position number 3 and 1 at position number 4. So, what it is, when we have seen this structural isomer, this is 1, 2, 3, 4 and 5. Again this is a pentanoic acid. The only difference is, rather being 2-propyl, now this time at position number 3 it has 2 methyl groups, and at position number 4 it contains 1 methyl group. So, it means it will contain 3 methyl group, 3, 3, 4. The position number 3 is occupied twice with the CH3 group, and position number 4 is also occupied with CH3 group, and thus it contains 3 CH3, the methyl group. So, it is tri means 3 and methyl group, and the same 1, 2, 3, 4, 5, though it contains 5 carbon atoms. So, it is pentanoic acid. So, this is 3, 3, 4 trimethyl pentanoic acid. These two, both the 2-propyl pentanoic acid and 3,3,4 trimethyl pentanoic acid, these both are active as anticonvulsant agents. So, now we will look at the structure activity relationship of valproic acid. So, as we talk about the anticonvulsant potential of these valproic acid derivatives, these anticonvulsant activity that gets increases with increase in the chain length. As the chain length increases, the activity also increases. Like suppose if we have 5 carbon atom chain, if we suppose and we get like we want to increase the chain as a substitute by putting some more substituent in the valproic acid, that will increase the activity. But when we introduce any double bond in this chain, that will lead to decrease in the activity. Apart from that, if we try to incorporate any secondary or tertiary alcohol group, or if we replace this carbonyl group or hydroxyl group, this OH group, this is OH, this is OH group, that will produce no effect on the activity. So, as it is, the structure the valproic acid will possess its anticonvulsant potential. So, again the structure activity relationship of valproic acid, the anticonvulsant activity that will get increases when we increase in chain length. So, if we suppose to increase this chain length as we have 5 carbon atoms in the straight chain, in the longest chain, if we increase the side chain, so that will increase the anticonvulsant potential. And suppose if we incorporate any kind of double bond, suppose say here, that will decrease the activity, that will decrease the anticonvulsant potential. Further, introduction of any secondary and tertiary alcohol group or replacement of carbonyl group or hydroxyl, that will not produce any kind of effect on these activity of valproic acid. So, let's go back and just revise the current lecture. So today we did two of the categories, one is benzodiazepine and another one is miscellaneous. So first one was the benzodiazepine. As we have discussed, it contains two of the rings, one is benzene, another one is diazepine, and in the diazepine we have a seven membered ring along with two nitrogen atoms. So chemically, if we talk about the benzodiazepine, it is a fusion of benzene ring along with a seven membered two nitrogen containing, that is diazepine ring. And then we have also studied these benzodiazepine act as anti-anxiety, also used in treatment of insomnia, apart from that anti-epileptic as well. They are very good at GABA enhancement, that the action of GABA receptor. Actually what happened, actually these benzodiazepine, they bind themselves to the allosteric site of GABA receptor, and they enhance the action of GABA. GABA, what actually GABA does, they will open the calcium, they will open the chloride channels. They will open the chloride channels and will inhibit the excitatory action of the body. So when GABA itself is producing calmness in the central nervous system and the benzodiazepines are supporting them, so in this way they are acting as in the treatment of anxiety, because you have so many thoughts and so many anxious feeling when you are suffering from anxiety, and insomnia, that the patient is not able to sleep. So in that case also benzodiazepine really works well. In case of seizures, when there is a extra nerve impulse action, when there is extra signaling from the nerves, so in that case also benzodiazepines work well. So these are the examples: diazepam, nitrazepam and clonazepam. This is the structure activity relationship of benzodiazepine. As we have discussed, we have four different targets in case of benzodiazepine. The first one is nitrogen at position number 1, that we have studied. Like if we do the N-methylation, that will definitely increase the activity. When we talk about the position number 5, that is essential for the activity, that the phenyl substitution must be there if we want benzodiazepine to act as anticonvulsant. When we talk about the position number R2 dash, R2 dash means the second position, the ortho position of phenyl ring, that is substitution at phenyl at position number 5. So the substitution at position number 5, we have R2 dash. So at R2 dash, there is if we have substituted this position with a halogen, that will increase the activity, but when we substitute this position with the electron donating group, that will definitely decrease the activity. Apart from that, we have position number 7. Position number 7 again having the same working profile as that of the R2 dash, that the electron withdrawing group, that will serve the purpose, increase the activity. Rather this is not encouraged to substitute the position number 7 with electron donating group, like any alkyl group, CH3 or OH group. Further, the miscellaneous agents, we do have primidone with 2 ketonic group with 2 NH group, valproic acid. Then the felbamate with the CONH2 group, and gabapentine with a carboxylic group. Further, we have studied about the structural isomers of valproic acid. Here we have studied, like we have 2-propyl pentanoic acid, because at the position number 2 we have this propyl group. So with this propyl group, 2-propyl pentanoic acid we have. Further, this propyl that contains 3 carbon atom, these 3 carbon atom in the structural isomer, they have been distributed at position number 3 with the 2-methyl group as 2-methyl group, and at position number 4 as single methyl group. So it contains, like the name suggests, 3,3,4 trimethyl pentanoic acid, and that is almost equivalent active as that of valproic acid. The structure activity relationship of valproic acid: when we talk about the structure activity relationship, the anticonvulsant activity will be increased if we increase the chain length. Like we have this 1 2 3 4 5 the main chain, and we have 3 carbon side chain. If we increase the number of carbon atom in these chains, that will definitely increase the anticonvulsant potential. But by the introduction of any double bond, if we incorporate double bond in this drug, valproic acid, that will decrease the activity, while introduction of any alcoholic group or any change in any carbonyl group or hydroxyl group, that will have no effect. So this is all about the structure activity relationship of valproic acid. So as we have completed all the categories of anticonvulsant as well, so hence this chapter is also completed. And so we have studied the structure activity relationship and as well as the synthesis of important drugs of the anticonvulsant potential. So this chapter is completed. Thank you very much.